Edward Taylor. He received his PhD in condensed matter physics in 2008 from the University of Toronto. And after completing his master's at Queen's University, he received his bachelor's degree from the Royal Institution for the Advancement of Learning in Physics. And after several postdocs in condensed matter physics in Italy, the US, and Canada, he entered the world of medical physics in 2015, becoming a postdoc with Dr. David Jeffrey at the Princess Margaret Centre. After completing a medical physics residency during 2019, he was hired as a staff member, where he has been ever since. And his clinical work has focused on palliative and head and neck cancers, as well as combined MR linear accelerator. And so welcome, Dr. Taylor. And the title of his topic today is going to be integrating hypoxia imaging into the clinical workflow to improve radiotherapy response. Welcome, and the floor is yours.
Thank you very much. The statement, by the way, that I got my Bachelor of Science from the Royal Institution for the Advancement of Learning was a slightly inside joke. I don't know how many in the audience recognize that. I'm a McGill undergrad, and the formal name of McGill University is the Royal Institution for the Advancement of Learning, not McGill. So, I already tried sharing my screen, so this should work. I'm just getting over a cold, so I think I have. I'm a little hoarse, so hopefully I can get through this talk. It's not terribly long. It should be done in 35 to 40 minutes, hopefully. But feel free to interject and raise your hand at any point if something's not entirely clear. I'm also going to take lots of breaks for water. So I've spent the bulk of my career in medical physics thinking and looking about hypoxia, at least the research part of my career. And one thing that I'm very aware of is that hypoxia is everywhere in the literature. People have studied it ad nauseum. There are many research studies on hypoxia, and it's well known that hypoxia negatively impacts radiotherapy, which is of particular interest to me as a medical physicist. And yet there's almost no routine strategies to ameliorate the impacts of hypoxia. So this was a paper written by Jens Overgaard, who's one of the deans of hypoxia research, quite some time ago in 2007. And he talks about this situation in this paper. He's referring mostly for hypoxia-activated drugs, not necessarily other potential adaptation scenarios that we could envision, but he talks about hypoxia radiosensitization, again, a drug-based viewpoint, adored and ignored. So it's adored because there's so many papers on hypoxia and the ways we could potentially modify hypoxia treatments, modify treatments, so standard chemorads, to overcome the impact of hypoxia, and yet nobody actually does this in clinical studies. Now, I think there's multiple reasons for that, but one of the most basic, which I'm really going to focus on in this talk today, is that our ways of actually measuring hypoxia are quite expensive and quite difficult to do in routine clinical workflow. And so really the leitmotif of this talk is trying to develop a new way of bringing hypoxia imaging into the routine clinical workflow. So just briefly, hypoxia is a state of low oxygen that often exists in solid tumors. So tumors often have a dysregulated vasculature as compared to normal tissues. And so if you picture, as is shown on the left-hand side of the screen, an oxygen gradient sort of as you move further away from capillaries, because of the disorganized vasculature in a tumor, you are more likely to find regions where you're further away from capillary, too far for oxygen to diffuse, and so because it's consumed at the same time, and so you have regions of low oxygen, and that's also what you see on the right. Is an immunohistochemical stained image of blood vessels. So those are the red images. Also perfusion, because not all blood vessels and tumors are perfused. So that's blue. And then the green is a marker for hypoxia called EF5. And so what you see is that in regions that are far away, far meaning typically between 80 and 150 microns, regions that are far away from perfused blood vessels develop hypoxia. And so if you were to apply, look at a similar image such as this for typical normal tissue, you would still see the odd hypoxic subregion, but they would be volumetrically much smaller. Here we see quite a bit of hypoxia. So the key takeaway points for hypoxia: it exists to some degree in almost all solid tumors. That being said, the magnitude of hypoxia does vary quite a bit from tumor to tumor. And not only does it vary from patient to patient, tumor to tumor, it varies in time. So we know it's a temporal phenomenon. It can vary during the course of radiotherapy, and reoxygenation is one of the critical factors needed for conventionally fractionated radiotherapy to be successful. But even we know that there are, or at least it's suspected that there are, shorter term transient hypoxia occurring on timescales of 30 minutes to an hour. So in terms of the impact of hypoxia, the most direct one, of course, is that oxygen is a radiosensitizer for low LET radiation, which is most of the radiation we use in the clinic. Oxygen is needed to render hydroxyl-induced DNA damage permanent. And so without oxygen, the impact of radiotherapy is decreased by almost a factor of 3. But then in addition to that, it also contributes to genomic instability and phenotypic diversity, because cancer cells that living under a hypoxic environment need to try and gain some evolutionary advantage, so they try and adapt to a new phenotype. It also selects for clinically aggressive, more metastatic behavior. It indeed increases metastatic potential, and it contributes to treatment resistance, not just radiotherapy, but also chemotherapy. So this is a sort of a classic study on hypoxia and cervical cancer. And there should be a third image there, but it's missing. It's not that everything's shifted over to the left. On the left, you see disease-free survival. You see there's quite a big difference between the two curves. So these are cervical cancer patients who underwent some hypoxia assay. In this case, it was polarigraphic probe measurements. And so these researchers distinguished two different populations, those that they labeled as hypoxic, meaning those for whom the percentage of polarigraphic probe measurements which revealed a local oxygen tension less than 5 millimeters of mercury, that was deemed to be hypoxic, and another cohort of patients for whom in less than 50% of the cases, the measurements were above 5 millimeters of mercury. And so just using that binary classification scheme, they're able to see a substantial difference in disease-free survival between those two cohorts. Local failure, there's less of an impact, but it's still there. And what the third picture would have shown you was that metastatic, in terms of metastatic dissemination, the hypoxic patients metastasize much more, which is why they have much worse disease-free survival than the non-hypoxic patients. Oh, there it is. Sorry. Okay. The same scenario recapitulates itself in other tumor sites as well. These are head and neck patients, two different studies. On the left, this is PET imaging of hypoxia, F-MISO-PET. And they're looking at local regional control in 35 head and neck cancer patients. This is hypoxic versus non-hypoxic. Again, the hypoxic patients do quite poorly. The non-hypoxic patients or oxic patients do quite well. This is just standard in response to standard chemo rads. And the study on the right is sort of interesting because they looked at actual follow-up measurements. So these are all 70 gray and 35 fraction head and neck cancer patients. So they took, in addition to a baseline F-MISO PET imaging, they took a follow-up F-MISO PET image 2 weeks into treatment. And what they actually found was that image set 2 weeks into treatment was more predictive of local regional control than the baseline or at any other time point. So the conclusion there was that not only is hypoxia important, but as one might expect for conventionally fractionated radiotherapy, the kinetics of hypoxia are important. So it doesn't really matter what the upfront hypoxic status of the tumor is. For these patients who undergo 6 weeks of radiotherapy, if a lot of that hypoxia is resolved after 2 weeks, then they're going to do well. If it doesn't, then they do poorly. I'm just glossing over so far what these sort of different means of measuring hypoxia are. I talked about polarographic probes on the previous slide. This is something called F-MISO PET. That's a niche that's a PET modality for imaging hypoxia. I'm going to provide some more details of that in a bit. And arguably the most important thing when it comes to thinking about hypoxia-targeted therapies is the notion that we need to stratify patients for these therapies and these studies. So this is just one example. It's 861 head and neck cancer patients who were randomized to chemo-rad, standard of care chemo-rad, plus or minus tirapazamine. So tirapazamine is a class of drugs is a member of a class of drugs called hypoxia-activated prodrugs. These are drugs that bind to cancer cells, or actually any cells, or they're not like most chemo drugs. They're not very specific, but they bind to DNA in cells under low oxygen conditions. So these are what we call hypoxia-activated prodrugs. They're drugs that are attracted to hypoxic regions of cells and only work under hypoxic conditions. And 861 head and neck cancer patients, and really no statistically significant difference between tirapazamine and just standard of care chemo-rads, which is here it's indicated CIS, that's just cisplatin plus radiotherapy. So from this, and then studies like this were common to hypoxia-activated or hypoxia treatment. So there was a lot of excitement in the early noughts about developing these drugs, pushed by pharma companies, and virtually all of them came up with results like this, which were essentially negative. So this was disappointing. However, there's two critical takeaways here. One is that radiotherapy plan quality is crucial. So this is not relevant at all to hypoxia, but as a medical physicist, I feel obliged to point this out. 25% of the plans in this study were non-compliant. So specifically, they didn't meet a particular set of clinical goals that the study identified as being crucial. And on secondary review, 47% of these 25% non-compliant plans were found to have major impact on tumor control. And so in the upper right-hand side of this slide, it's showing the percent survivors. So this has nothing to do with tirapazamine. This is both cohorts of patients, the percent of surviving patients for four different cohorts of radiotherapy plan non-compliance. And so in particular, they identified the major TCP impact patients were the ones for whom they felt they had a major impact on tumor control. So for instance, you could have a plan where carotid dose was higher than what the study guidelines indicated, but you could also have another plan where PTV was not covered by 98% of the PTV wasn't covered by 95% of the prescription dose. So one constitutes major, one has a major impact on tumor control, one does not. And so identifying the ones that do have a major impact on tumor control, they found that those patients did much worse. And you see it's not even close. It's about, it is about 20, 20 to 25% difference in patient survival based on those two cohorts. So what that's telling you is even if tirapazamine has a strong impact on tumor control, and this is true of any randomized control study that involves radiotherapy, radiotherapy compliance is critical because it can easily wash out, noise in radiotherapy compliance can easily wash out any signal from your drug. And so this is one of the major reasons why I always tell my radiation oncology colleagues it's so important to have protocols, so important to have physics review of protocols. The other thing, so once they sort of filtered out the non-compliant plans, then they found tirapazamine sort of edging towards statistical significance. So remember on the previous slide, the p-value for tirapazamine outperforming cisplatin was about 0.67 and now goes down to 0.06. So it's getting close to statistical significance here. Still not quite there. The final thing you need to put you over the edge, though, is patient stratification based on hypoxic status. So as I mentioned before, in a large patient cohort, there's going to be a large variation of hypoxia. And so in a smallish 45 patient sub-study of this larger study, 45 patients received F-MISO PETs, and that's a PET imaging modality of hypoxia to determine hypoxic status. The rest of the patients, they had no idea. Okay, everyone was randomized to this hypoxia-activated prodrug or not, without any knowledge of what their hypoxic status was. And so what they found is amongst these 45 patients, if they stratified using this PET hypoxia metric based on hypoxic or non-hypoxic, then there's a huge difference, at least for tirapazamine. So the patients who were hypoxic and didn't have tirapazamine had about a less than 30% of them did not have local failure. Conversely, of the hypoxic patients who did have tirapazamine, almost 100% did not have local failure. So it's a huge difference. On the right-hand side, just to give you an example of what these images look like, these are taken from this study. So it's showing you an FDG baseline image, and you can see the two kind of hot areas of FDG. One of those is a node, and one of them is a primary. So the more medial one, smaller volume, is a primary. And then right below that, you see the F-MISO PET image. What the F-MISO PET image reveals is that it's, the primary is fairly oxic. There's no hypoxia, but the node is very hypoxic. 12 weeks later, after radiotherapy, the FDG is repeated. You see the nodal volume is still quite FDG avid, which indicates it's still there. It hasn't really responded to radiotherapy, and sure enough, that was the hypoxic sublesion. The primary volume is gone on FDG, and that's because presumably it was oxic, and so the oxic tumors responded very well to treatment. Okay, so just a word about PET imaging of hypoxia. As I'm sure most of you know, PET imaging is widely used in oncology, but it's at a 99.9% level. It's fluorodeoxyglucose, FDG PET. There are niche sort of tracers that are can be used for other applications. So one class of compounds are so-called nitromidazoles. These are originally compounds developed as to combat bacteria. They were, I can't remember the name, but they were antibacterial compounds developed in the 1950s. And it was discovered that they bind to large macromolecules under low oxygen conditions. And so these were converted to a class of compounds such as F-MISO is by far the most well-known. Also FASA, which is one we use at Princess Margaret. These are compounds that can detect hypoxia via PET imaging. And on the right, you see some example images. So on the top, you have a cervical cancer patient. It's right below. This is an example of a hypoxic tumor, and this is a very substantial signal. You see a hypoxic tumor just below the bladder, so the bladder is really bright there. And just below that is a pancreatic cancer patient tumor. This is very hypoxic. This example, it doesn't look terribly hypoxic. That's because these imaging agents, different than FDG, they tend to have relatively low sensitivity, but that's a very hypoxic tumor. So these are compounds that are injected into the blood before imaging, and so they make their way through the entire body, but tumors as well, by passive transport, so extravasation, and then diffusion. They're freely diffusible molecules that they can cross the cell membrane. And then if they encounter low oxygen environments inside the cell, they don't undergo one electron reduction reaction, and they remain in the cell membrane. So the advantage of this approach are principally it's minimally invasive. So before PET hypoxia imaging, and this is about a 30 to 40-year-old imaging modality, by the way. It was really developed at the University of Washington in, say, the early 1990s. And so the alternative before that were polarographic probes, which is literally a needle that is just pseudo-randomly placed inside a tumor that's relatively near to the surface. It's very sensitive to technique, and very few people do this anymore, but that was kind of the way you do that before. The advantages in comparison of PET hypoxia imaging are it's minimally invasive, it's applicable. You can image the whole tumor, and it's repeatable, so you can repeat it after a few weeks. The challenges are limited spatial resolution. This is PET. It tends to be 4mm voxel sizes. You get uptake in normal tissues. The sensitivity is relatively low. There was a lot of controversy early on about whether or not it's better to do a static image, which is what we tend to do in FDG PET, for instance, versus acquire a dynamic image as well. And then there was still uncertainty in how to analyze and report data. What is the best metric for hypoxia imaging? We don't, as a general rule, use SUV, for instance. That's the standard FDG metric. We use hypoxic fraction, which just indicates the fraction of voxels, image voxels, that are greater than some threshold. Excuse me. This was actually a slide deck that I had about 8 years ago, and I decided this list, the advantages and challenges, was a list I had 8 years ago. I decided to update it, having been a working medical physicist for the past 8 years or so. I now would say the biggest challenges by far are low sensitivity. That remains constant, but it's also really expensive. So we acquire these PET images on research study basis, on a basis of research studies. It costs us about $5,000 scanner time plus the phase generation, and it's time consuming. It takes about an hour per imaging session. It actually takes us longer because we still acquire a dynamic image. So we acquire a half-hour dynamic image for our patients, and then we get them they get off the treatment bed. They come back 2 hours later to allow full biodistribution of the drug, and then they're imaged for another 40-minute session, which is just what we call a static imaging session. So even in the simplest scenario, though, these drugs are injected, they require 2 hours to properly biodistribute, and then there's a 45-minute imaging acquisition. So there's absolutely no way that you can scale this process. You're never going to get a substantial portion of patients to undergo this type of imaging. So no matter how good this imaging is, no matter how much you might like it, it's fundamentally limited in terms of applying this to all patients. And of course, we would like to do this for all patients, and also randomized control trials. So remember the Head Start trial I showed you in the first few slides, 861 patients. There's no way you're going to be able to image all 861 patients or stratify them all for hypoxic status using PET imaging. Just too expensive, too time consuming. So we need alternatives. So there's actually a number of ways to image hypoxia. The classic one are these polar graphic probes. And that's, I would differentiate three different kind of areas of hypoxia detection. On the first column, I would distinguish what I call direct probes of oxygen level. The middle column, I have indirect probes of oxygen. So that's some kind of indirect probe that uses a model to deduce the level of oxygen in the tumor. And on the third one, I have what I call probes of radiological hypoxia. So that's assessing what are the kind of radiobiological, what are the radiobiological signatures of hypoxia, and can those be detected? So kind of downstream factors. And so coming to direct probes of oxygenation, we have our polographic probes, which, as I said, are almost never used. They were only really applicable to tumors that were near the surface. They're very operator dependent, and it's very timely. There's also paramagnetic electron spin resonance, which benefits from the fact that oxygen is a very powerful spin relaxation contributor to spin relaxation for electrons, paramagnetic substances. We don't do a lot of paramagnetic electron spin resonance, but I feel obliged to mention that one. There's also tissue or oxygen-enhanced MRI, which is a really exciting possibility, and I've bold-fonted here because I'm going to discuss it a little more on the following slides. Then there are indirect probes of oxygenation. So bold MRI is a kind of a classic one. It is sensitive to relaxation due to differential hemoglobin occupancy, oxygen occupancy levels. Because it's a long way from hemoglobin to hypoxic regions and tissues, that's only a very indirect measure of oxygen or hypoxia in tumors. So people have looked at that. I would say it's pretty much a dead story. It's not useful for hypoxia detection in cancers. You can look for perfusion measurements. Since I said at the beginning, hypoxia is a result of poorer perfusion that tends to arise in tumors. And so people have looked at dynamic contrast enhanced MR and also dynamic contrast enhanced CT surrogates for hypoxia. Those, again, have not largely panned out. A third very interesting option, which has been the basis of my research, is diffusion-weighted MR surrogates of hypoxia. I'm going to talk about those in the next few slides. We'll also mention these radiobiological signatures. So in addition to PET, which I consider to be a radiobiological measure of hypoxia because it involves compounds binding to macromolecules, principally DNA, under hypoxic conditions. So radiobiologically significant hypoxic conditions. You also have downstream markers of hypoxia, such as hypoxia-inducible factor, HIF, GLUT, which is glucose upregulated transport, VEGF. These are all upregulated in response to hypoxia. And then correspondingly, you can have genetic signatures of hypoxia. So these have been used to varying degrees. They're also problematic. They're not perfect correlations, and they are sometimes difficult to implement longitudinally. So my focus has been on these two MR-based methodologies, which I'm going to discuss next. Okay. Oxygen-enhanced MR is a very interesting idea. It first sometimes called tissue oxygen level dependent MR, told to distinguish it from bold. The basic idea is illustrated in this plot here. So a patient would get a T2 weighted anatomical image, kind of standard. And then there's a T1 acquisition over time as the patient initially is just breathing air, and then at some point the patient begins to breathe oxygen. And so what is of interest here is how does the tissue signal change, in particular the T1 relaxation, how does a change in response to oxygen breathing? So oxygen enhances spin decay. And so the idea is that if I have a tissue that does not respond, that has it's kind of refractory to this T1 change, then it means there was no oxygen there to begin with. This is a hypothesis. Conversely, if I have a high level of oxygen in my tissue, it's well oxygenated, then the difference before and after between breathing oxygen will make little difference on T1. And so doing this, the researchers that have put forth this method hypothesize, and this has been validated in preclinical studies, they hypothesize that voxels that are refractory on T1 to oxygen breathing and are those that are hypoxic. And so on the left, you see a sort of a pseudo image of a hypoxia in a primary head and neck cancer patient. And the idea then is the blue regions are the oxygen refractory voxels, and so those represent the nominally hypoxic regions. I should say this has never been validated against a sort of
Yeah, we've lost the speaker. See pause for everyone else. Yeah. Hopefully it comes. Oh. Let's see what happens. Oh. Oh.
that was me.
Okay.
I actually have very bad Wi-Fi in my office. That was okay. I need that break anyhow.
All right, we can pick it up again.
Okay. What I was saying was, I've forgotten what I was saying, but these are images acquired on a combined MR magnetic resonance.
Your slides disappeared.
Very good. I didn't share them. Okay.
Good.
Thank you. Apologies for that. So this was this particular workflow was developed for a combined magnetic resonance linear accelerator, combined MR linear accelerator, MR Linac. And so the exciting thing with this is you could in principle do this for a large volume of patients. The whole acquisition takes about 10 minutes. You could do this for your patients and that are undergoing radiotherapy treatment on MR Linac, and you could actually in principle use the data from this oxygen-enhanced MR sequence to inform your treatment. You could deliver elevated boosts to the hypoxic subvolume in theory. In practice, this machine does not allow you to do that just yet. So you can acquire the images and you can accumulate the data, but we can't actually act upon it just yet. That may change in the coming years. So we actually very recently just did this to our MR Linac. So this is just about 6 weeks old, this photo. And we had to take it out of service for a week. It was a lot of work just to install a bunch of pipes. But in that red ellipse there, you see the oxygen taps, which will allow us to do this. In fact, we are now able to do it. We just haven't done it yet, but we now have the capacity to do this too. And so we hope to start acquiring these images fairly soon. The other methodology I mentioned was diffusion-weighted MRI as a potential hypoxia surrogate. And this is the one that I've really been working on for a long time. It doesn't offer any real advantage over the oxygen-enhanced MR imaging I showed you on the previous page, except for the fact that it can probably be done a little bit faster, and diffusion-weighted imaging is a very widespread technique. And so in principle, this is highly scalable. But the downside of a diffusion-weighted MR hypoxia circuit is it's very model dependent. So it depends on making some models of that model of how hypoxia arises, and then the model being correct. So speaking of models, so it is as kind of a mechanistic point of view, hypoxia is a relatively simple phenomenon. It arises from the interplay between oxygen supply, which is the blood vessels, the vasculature, which supply the oxygen, and oxygen demand, which is the oxygen consumption rate, so the rate at which tissue utilizes oxygen. And so if you have, a poor vasculature or low oxygen supply, and, you're going to get hypoxia unless you have really low oxygen consumption. Conversely, if you have high oxygen consumption, then you need a really, really strong supply of oxygen in order to not have hypoxia. And so hypoxia is fundamentally a really simple thing to understand. The problem is that neither of these two variables, either oxygen consumption rate or oxygen supply, are directly measurable. Probably the closest we come to is oxygen supply. We can measure blood vessels using the presence of blood vessels using a number of techniques, including DCE, CT, or MR. But what's of particular interest to me with my DWI hat on is intervoxel incoherent motion. That can tell us that can quantify the presence of the vasculature. And in terms of oxygen consumption rate, a 0th order approximation is that the oxygen consumption rate is proportional to the density of cells. So if I have relatively acellular tissue, it's going to have a low oxygen consumption rate. Conversely, if I have a lot of cells jammed into a tight space, it's going to have a high oxygen consumption rate. At least that's a 0th order approximation. So just a word on intervoxel incoherent motion. This is a kind of in the middle, you'll see a classic logarithm of the signal from a diffusion-weighted MR sequence. And, what we do is we tend to fit that kind of middle range, the area known as B. We fit that middle range, we just do a linear fit, and that's our apparent diffusion coefficient, which is inversely proportional to cellularity. So we know that if we have a deeply cellular tissue, it's going to have a low ADC value, which is a major prognostic imaging metric. The ADC imaging is used in many different cancers. But then there's this sort of characteristic upturn that always shows up at low B values. The B value here, which is the ordinate along the x-axis, is just basically the magnitude of the gradient of the fields used to generate these images. And so typically these, for diffusion-weighted sequence, you acquire images over a number of B values. And what people notice at small B values rather, is this upturn in signal. And so for a long time, people have been advocating just use a signal for B values between 150, 200, and depending on your scanner, 1,000, to get your ADC value, because we know that there are fusion effects that determine that low B value signal. And it was Dennis Lebehan in the 1990s who sort of proposed a model that quantify this. The bi-exponential model is shown on the right, and the idea being that the amplitude or magnitude of this upturn at low B values is proportional to the fraction of the tissue that's occupied by blood vessel. And so the qualitative picture that he envisioned was that blood moving through tortuous vessels looks a lot like kind of a pseudo diffusion process. And so it's characterized by a fast diffusion, which he called D star. And it also allows us to extract the blood volume fraction. And so IVIM is just kind of an extended diffusion weighted scheme. We just add some p values, and it allows us to get not only the ADC, which is proportional to inversely proportional to the cellularity, but also the blood volume fraction. Until I'm running out of steam, I'm no longer able to talk much. So this was done by Tord Homplin and colleagues at looking at prostate cancer. And so they used this IVAM plus, the F, they call it FPV here, the blood volume fraction, plus ADC imaging to distinguish hypoxia from non-hypoxic patients. They had the advantage, these were done for patients who were undergoing resection. So they had pymanidazole, which is a metric of hypoxia, immunohistochemical metric of hypoxia, and they were able to validate it. They had beautiful results. So this is showing you on the left. This is quite a hypoxic patient. The brown on the left on the histology image is pymanidazole. The false color image on both panels are these hypoxic kind of maps that are determined by ADC and F values. And so again, low, what they just decided, you can see that on the far left-hand plot here, they identified hypoxic patients as, anyone who had, or a hypoxic voxel as being hypoxic if, the F was less than some critical value. So, and also ADC was less than some value. So hypoxia being associated with dense tissue, but also poorly perfused. And so the end result is shown on the right here. They had a very high R value, very strong predictive capacity to identify hypoxic patients. So I thought this was great. We should do this at Princess Margaret. And for a variety of obscure reasons, we ended up doing this for soft tissue sarcomas and also pancreatic cancers. Pancreatic cancer is the one that I've spent most of my time analyzing, so I'll discuss that. And these were also tied in with patient treatments. And so patients would get a combined FASA-PET image and diffusion-weighted image on a combined PET-MR scanner. This was within 1 week of their start of treatment. They would go to the Elekta or MR Linac for treatment. This had nothing to do with the development of the hypoxia biomarker, except we did want to test if we could, if we had a biomarker, if we could apply it to, or if it were extendable to an MR Linac. So patients underwent 40 gray in 5 fractions. We did also add a hypoxic boost for hypoxic patients, but basically 5 fractions every second day. Every fraction we acquired diffusion-weighted imaging. So if we were able to acquire, deduce a hypoxia-sensitive diffusion-weighted imaging surrogate of hypoxia, then we could also track that through treatment using our MR Linac device. And then after treatment was done, within a week of treatment ending, patients would go for a repeat imaging session on our PET-MR imaging device. And so we'd have a data set which allowed us to develop a model for hypoxia because we had the FASA-PET and the diffusion-weighted imaging overlaid. So we could develop a model for hypoxia-based MR surrogate, and then we could, in principle, track it through treatment and then see it on the other side. Also, we wanted to know how much did hypoxia change. So these are relatively important treatment times. These are 5 fractions within 10 to 12 days. So we wanted to know, does any meaningful reoxygenation occur during that time? And so this is just our initial data. This is showing the kind of the same analysis that was done in the publication I discussed a couple slides ago for prostate, just using a very simple metric for hypoxia based on diffusion-weighted imaging, compared with the hypoxic signal we got from PET. It's actually a pretty good signal, R is equal to 0.65. I wouldn't complain too much about that, except that looking at in detail, what you'll see is that a lot of that correlation is driven by patients who are very hypoxic. So they have high signal on FASA-PET, And really where most of the noise comes in is at the low hypoxia level, which is problematic if you think about stratifying patients for hypoxia-targeted therapies, because really it's patients we know from an abundance of clinical evidence that the threshold for what constitutes clinically relevant level of hypoxia depends on cancer, depends on treatment, but it's towards the lower side of this plot. So meaning pretty much any patient with hypoxic signal here, it's hypoxic fraction less than 20%, maybe even less than 10%, those are still, we would consider them still to be hypoxic. It's only really the patients who have almost no hypoxia, at least on PET. Remember that PET is a low sensitivity methodology. It's only patients with really low hypoxic signal that we tend to see a potential benefit from dose de-escalation or non-stratification for hypoxia-targeted therapies. So really, this from a stratification point of view, although strong correlation, is driven by very hypoxic patients, and we would like something that has better sensitivity, especially at low levels of hypoxia. So one qualitative observation I made in looking at this data was that the patients who were very hypoxic were almost always dense, poorly perfused. That was which I would expect. But then so those were very easy to stratify if you saw them. But most of the patients lived in this kind of intermediate regime where there's lower density, variable perfusion. It varies from patient to patient. There's no clear rhyme or reason, and yet they can still be, quite hypoxic. And so the assumption that I went back to was the assumption that, of constant, that the oxygen consumption rate was proportional to the density of cells. So that assumption is basically saying every cell has the same level of oxygen consumption. And of course, we know that's not true. So studies have demonstrated that different cells consume oxygen, different cancer cells, even of the same cancer type. This is pancreatic cancer, consume oxygen at different rates. The reason for that is because we know the cancer cells, they start off using oxidative phosphorylation, and over time they evolve towards a phenotype where they metabolize glucose anaerobically, producing lactic acid, sometimes called the Warburg phenotype. And so depending on where you catch the tumor in this evolutionary step, you might find that, your cellular density is not a good metric, or is not very sensitive to oxygen consumption rate. You could have cells, for instance, in principle, if they relied completely on anaerobic glycolysis, didn't use oxygen at all, they could be deeply cellular, very limited perfusion, and still be low levels of hypoxia. And so that led me to develop another model, which I'm not going to spend much time discussing, which is a shame because it's much more complicated. But the idea was to use diffusion-weighted imaging to see if we could and fit a model for tumor growth and evolution, fit that model to determine whether the extent to which a tumor engaged anaerobic glycolysis. And so this is on the right-hand side, you're looking at a plot of, this is IVAM data. You're looking at a plot of the cellular density. So that's basically 1 upon the apparent diffusion coefficient versus the vascular density, which I also get from IVIM. And so this is what I call a classic non-hypoxic. This was an oxic tumor. And so you see there's a strong correlation between density and perfusion, which makes sense because the more blood I have in a given region of tissue, I expect that to support more cells, and so the cells grow there. And so in my model, this corresponds to a completely oxic, non-hypoxic tumor because because there's no signature of glycolysis. This is another model of a tumor which is extremely hypoxic, and what you find is in that very hypoxic region, there's a region of high density but low perfusion, which the model identifies as a glycolytic, anaerobically glycolytic phenotype. So these are cells that have evolved towards glycolysis. They're still hypoxic because they grew out of a hypoxic environment to evolve to this phenotype. And so this is the modeling now. And when you incorporate that level of modeling, and it should be noted, only adds one extra fitting parameter, the correlations increase quite a bit. And notably, we have much better sensitivity and specificity at low oxygen or low PET, low signal levels. Okay, just one final screenshot just showing you this in action. So on the left-hand side, you see two example positive PET images of two tumors, one that's hypoxic, one that's not. On the second panel, second column, you see the model that uses the diffusion-weighted images acquired at the same image set to predict hypoxia. And so the top tumor is correctly predicted as being hypoxic, the bottom one is not. And then we wanted to see if we could also implement this biomarker on our MR-Linac. And so we have basically the same model applied to the diffusion-weighted images acquired on the MR-Linac. And at the first fraction, we see hypoxia there, very similar in the first patient, but on the other. The interesting thing is that by the fifth fraction, that hypoxia seemed to have resolved, which was in fact demonstrated on the follow-up PET-MR sequence. So we have a nice, potentially nice, I'll actually just, rather than leave it summary there, I'll just leave it here because it's a nicer thing to look at. So we have preliminarily, we have a potentially useful biomarker for hypoxia and tumors. The very nice thing is it does seem to be reproducible. So not only can we apply it to our fancy PET-MR, it seems to also work on our MR-Linac, and that opens the door to acquiring diffusion-weighted imaging and signatures of hypoxia for every patient we treat on this, not just on our MR-Linac, but almost any patient who undergoes radiotherapy treatment and also gets an MR scan. So my hope is that every patient we won't have validation anymore because the FASA part was very expensive. But my hope is, and we're starting to do this, is we can acquire IVIM images of every patient undergoing radiotherapy through treatment. And, in particular for the patients on our MR-Linac, they'll get these sequences every day, and we can start to collect this data and look at treatment response as our surrogate measure for hypoxia. Okay, thank you very much. And thank you for bearing with my illness. Happy to take any questions.
Thank you very much for the very interesting talk. So now we will have a few minutes for questions from the audience. So I see that there's already a lot of people wanting to ask questions. It was very interesting. Myself, I have some questions, but I'll give priority to the students. So is there anyone here who wants to ask something? If not, we will go with Jorge.
Sure. Thank you so much, Dr. Taylor. Really interesting presentation. I have so many questions. So if you indulge me, I will have only two. I will limit myself to two. So number one is, I really appreciate the fact that you differentiated techniques based in basically which aspects of hypoxia do this technique interrogate. For example, TOL versus BOL versus PET-based techniques. Now, given this intrinsic difference, and when you compare techniques, PET-based techniques versus MRI techniques, how much of the difference that you are seeing can we attribute to the inner difference between interrogating cellular level hypoxia versus more of whatever metric will give you the MR-based technique?
Can you, I think I understood the question. Can you repeat it?
Sure.
Although you're asking what is the, if I just take
So, for
Say
example,
told,
if we go back to your plot that you compare F-MISO-based techniques versus your measurements in DCMRI, the linear plots, you know,
oh, this one?
the one in the next slides, F-MISO,
Sorry.
that one, for example, you are seeing a difference
Okay.
between those techniques. Like the slope is not perfectly one, and that difference will be at some point due to the difference between PET-based techniques versus MR-based techniques. So how can we
Yes.
account for those differences? How can we validate our MR-based techniques if there is going to be always an intrinsic difference?
Yeah, it's I mean, that's the critical question. So first of all, this is a fit, right? So we
Sorry.
so the y-axis is this hypoxic fraction, which is literally just the number of voxels in a tumor for which the uptake of FASA in that voxel is greater than some threshold divided by the total number of voxels in that tumor. That's just kind of a very standard metric that people use. And then the diffusion weighted signal is fit to optimize the correlation between that signal. So the slope should be on the order of 1, but it's a fake, it's a completely fake comparison because it's a calibrated signal. So at the end of the day, we do want to identify. I mean, it may be that hypoxic fraction is not a meaningful metric. It comes from old polographic studies where people would find that, if 25% of your measurements were, or 25% of the tumor was low oxygen, those patients did more poorly than the patients for whom only 10% were. So, as opposed to FTG, where we tend to use SUV mean and SUV max. So even hypoxic fraction is a particular metric that we use. It does represent, or at least it should represent in theory, the fraction of a tumor that exists at low oxygen levels. And so our diffusion, our MR metric should reproduce that. The catch is, of course, is that an imaging voxel is, encompasses many blood vessels, and it's a guarantee, it's a certainty that within any imaging voxel, you will have regions where the tissue is very well oxygenated, right next to a capillary, and then other regions where there's hypoxia. And so any measure of hypoxia, and this is true of PET and diffusion-weighted imaging based, is going to involve some averaging over this voxel, and it's invariably going to miss sort of occult hypoxia that's always going to exist there. It just may be too small in volume to manifest itself at the level of a voxel. And so, yeah, there's an apples and oranges comparison here, but ideally these are both telling us something about the level of oxygen in a tissue.
Yeah, that is great, and that talks volume about how difficult it is for MR-based techniques to be validated for hypoxia imaging and tissue oxygenation. And your answer was a perfect segue to my next question, in the sense that if hypoxia fraction is not the best metric, maybe, in quotes, what other metrics do you envision that we can use to do this type of comparisons or validations?
So I didn't mean to suggest it's not the best metric. It's just a metric that's being used. It's pretty good. So there are actually a few early studies that do use SUV max and have shown that has predictive capacity. In the future, hopefully, all this data will just filter all the data into a giant machine learning algorithm, and we won't have to worry about individual metrics. It'll just tell us that patients with this distribution of values for diffusion weighted or PET imaging tend to do poorly. So for us right now, we have to come up with a single biomarker metric, and hypoxic fractions of 1, but I can certainly envision a future where that's not the case.
Okay. Thank you so much.
Yeah. I'll read out Eve's question in the chat. Hi, thanks for an interesting presentation. Can you discuss the potential mechanism relating diffusion/IVIM to hypoxia? Is it physical relation or perhaps biological relation?
Yeah, so hopefully, I mean, I would say both. I did go through that quite quickly, but the idea is sort of encapsulated in this plot, which is so simple that it kind of has to be true, or at least it's very simple and is true. So hypoxia, can come about in a couple of ways, but really, if you have a poor, like the vasculature is too low in density, you don't have enough blood vessels, then, if you have a lot of cells consuming oxygen, then you're going to get hypoxia, and so the IVIM it tells us the blood volume density. So, it tells us what regions of the tissue are well perfused and which aren't. And then, so that's a direct physical connection. And then we use, the density. We take the ADC to be a surrogate of density. As I did explain, I didn't explain, but I just sort of very, very quickly went through it. What we found was that simple model of ADC being a surrogate for oxygen consumption rate didn't work particularly well, we felt. And so I came up with a much more elaborate model, but still only introduced one additional metric, fitting parameter, which is good. And that model, which is not you will not get anything out of these plots, or these equations. But the idea is that we are able to kind of, model the oxygen consumption rate by looking at these density versus perfusion plots. So these plots are kind of the basis of this analysis. So if I look at a case like this where density is very well correlated with perfusion, that's within my model, that's telling me that, okay, this tumor is kind of not really relying on glycolysis. This is just all the cells are breathing oxygen, so they all have relatively high oxygen consumption rate because they're following this predicted density is proportional to the amount of blood vessels relationship. When I see that breaking down, that tells me that glycolysis has started to occur. And again, here, cellular density is coming from the ADC, vascular density is coming from the IVIM. So all this is kind of a mathematically biological model interpretation of this density versus perfusion map, which is purely physical.
I have one question. So when you have done the glycolysis measurement or with imaging, have you seen any differences before radiation and after radiation? Do you see an activation of glycolysis after radiation, or it has been more like oxygen consumption for oxidative phosphorylation?
The glycolysis here is very theoretical level. We did have a couple of FDG scans for validation, but really this is just theoretical. Our endpoint is the PHASA-PET image, and that's what we're calibrating against. So what we did see is a reduction in PHASA-PET for some patients, but not all. So again, highly variable patterns, which is not entirely unexpected. So we imaged about 20 patients. Of the patients, it showed substantial upfront hypoxia on the PET image. I would say about half of them had a substantial reduction in hypoxia by the end.
Okay. Yeah.
Following up on Eve's question, he asks, is the model cell density, vascular density published somewhere?
Oh. No, I was still trying. Not still trying to get this published, still trying to write this. So I hopefully in the next 3 to 4 months, but I also said that 3 to 4 months ago. But we're looking to do that soon. We haven't published anything on this yet.
Great, thanks.
Thank you very much. And we will, I probably would like to contact you for collaboration if you're open for collaborations.
Absolutely. Feel free to reach out. I don't know if you have my email address, but anyone, feel free to reach out with further questions, or I'm happy to discuss any of this. I know that a lot of people have done really exciting FMISO and DWI and other MR work at McGill. So I'd love to collaborate and chat with anyone and all comers.
I think if we want to improve radiotherapy, we need to target hypoxia. So I think it's very important what you're doing and what many other people are trying to do. So I think collaboration is essential for getting there.
Absolutely.
Thank you very much. It was very interesting, your talk, and we really enjoyed it.
Thank you.
Thank you.
Bye.